Hello, in this 3D lecture, we're going to be discussing the digestion and preceding absorption process that takes place in the body of protein molecules. Okay? Now, in order to understand the digestion and preceding absorption process, two terms need to be well understood. That being mechanical digestion as well as chemical digestion. Now, mechanical digestion occurs in the mouth, while chemical digestion occurs in the stomach as well as the small intestine. Okay? Now, mechanical digestion, also known as mastication or simply chewing, is necessary in the process of digestion in order to increase the surface area of food molecules for digestive enzymes to interact with the peptide bonds that are present within protein molecules. Okay? So chemical digestion, it does not occur in the mouth. Only mechanical digestion occurs in the mouth. Okay? And again, that is to increase the surface area of the food molecules in order for proteolytic enzymes to break the chemical bonds via hydrolysis. Okay? Now, proteins or polypeptides are sequences of individual amino acids. Okay? So say we have one amino acid, two amino acids, three amino acids. They're connected via something called peptide bonds. So essentially, it's like a chain link that connects the two. And chemical digestion breaks these peptide bonds via this process called hydrolysis. Now, put very simply, hydrolysis is the addition of a water molecule, H2O, that comes in and cleaves that peptide bond. Okay? So when it cleaves it, a hydroxyl or OH molecule essentially dissociates with this amino acid, and a hydrogen ion, right, what is left of that water molecule, dissociates to the other amino acid, breaking that peptide bond. And that starts to occur in the stomach. Okay? And put very simply, that is what hydrolysis is. It's the utilization of a water molecule to break a chemical bond. Now, once the protein, the food, enters the stomach, that's where chemical digestion is able to start. Okay? Now, the primary proteolytic enzyme that starts to break down proteins into smaller polypeptides is something called pepsinogen. Okay? Now, pepsinogen is released by cells in the stomach called chief cells. Now, in the stomach is a very acidic environment. The pH is very low. And a low pH enables pepsinogen to work at its greatest rate. As a matter of fact, the low pH cleaves pepsinogen into its active form called pepsin. Okay? Now, pepsin is what cleaves the amino acids via chemical digestion. It is what starts the process of breaking the chemical bonds. Okay? Now, the food then travels to the small intestine, where further digestion takes place. Now, there's three primary sections within the small intestine. One being the duodenum. That's where digestion in the small intestine primarily takes place. The duodenum is the first section of the small intestine. The second and third sections of the small intestine are the jejunum and the ileum, respectively. Now, the pancreas, once food enters the duodenum, the pancreas releases proteolytic enzymes to further break down these polypeptides into smaller components. The three primary proteolytic enzymes that are released from the pancreas include chymotrypsinogen, trypsinogen, as well as carboxypeptidase. Okay? Now, there are other proteolytic enzymes that are released in the small intestine to make these, what they're called zymogens. They convert them into their active form. Now, there's a proteolytic enzyme in the small intestine called enterokinase, which cleaves trypsinogen into its active form called trypsin. Trypsin helps further break down those polypeptides into smaller components, okay? Now trypsin, it also cleaves chymotrypsinogen into its active form called chymotrypsin. Now chymotrypsin cleaves polypeptides that are called aromatic amino acids. Now, aromatic amino acids, their role is kind of irrelevant to this conversation, but that's what chymotrypsin primarily does. It cleaves polypeptides at the aromatic amino acid structures, okay? Now, carboxypeptidase cleaves proteins at their carboxyl end, okay? And if you want more elaboration on that, you can YouTube the structure of amino acids if you so choose. Now, we discussed mechanical digestion occurring in the mouth. Chemical digestion starts to occur in the stomach into polypeptides, into smaller components. Once it enters the small intestine, these enzymes break these polypeptides into smaller components. Now, these smaller peptides interact with the enzymes at what are called the brush border. Now, these brush border enzymes, they're known as dipeptidases. So the three main ones are called maltase, sucrase, and lactase, okay? They break down these small peptides into individual amino acids as well as di and tripeptides. Now, once that takes place, the absorption process is able to occur. And that's what this diagram shows, okay? So this is a diagram of an enterocyte. Now, an entero simply refers to the intestines, okay? So, and cyte refers to cell. So, enterocyte, essentially digestive cell. And this is where the absorption process takes place, okay? Now, there's different mechanisms in which the enterocyte works in order to absorb amino acids as well as di and tripeptides. So the biggest thing to understand is this sodium-potassium pump. This transmembrane protein, it shuttles 3 sodium ions out of the cell and it brings in 2 potassium ions. Now, if you want to know specifically on how the sodium-potassium ATPase pump works, you can YouTube it, or you can look it up in your physiology textbook if you have one. So this ATPase pump, it establishes a concentration gradient. Molecules and ions naturally move from a high concentration to a low concentration, right? So with 3 sodium ions being shot out, there is a lower concentration of sodium in the inside of the enterocyte compared to the outside, okay? So on the apical side of the enterocyte, that's this side is the basolateral side. So on the apical side of this enterocyte, there is a sodium-dependent cotransporter for individual amino acids. So because there's a higher concentration of sodium outside the cell because of this, sodium comes in through this cotransporter, it's going down its concentration gradient, and it brings individual amino acids with it. That's how individual amino acids are absorbed. Now, there's also something called the sodium-hydrogen exchange protein, which is present on the apical side. Now, since there's a higher concentration of sodium outside the cell compared to inside, sodium is going down its concentration gradient into the cell, right? And this exchange protein, it shoots hydrogen out. So it brings it brings sodium in and then it shoots hydrogen out, okay? Now, since hydrogen is being shot out, there's a higher concentration of hydrogen outside the cell compared to inside the cell, right? So that is how di and tripeptides are absorbed. Now, a dipeptide is just 2 amino acids connected by a peptide bond, and a tripeptide is 3 amino acids connected by peptide bonds, right? That's intuitive. So, because hydrogen is higher on the lumen side, or on this apical side, this hydrogen-dependent cotransporter brings hydrogen ions down. It's going down its concentration gradient, and it brings these di and tripeptides with it. Now, within the enterocyte, these di and tripeptides are converted into individual amino acids, and they're then absorbed into the bloodstream on the basolateral side of the enterocyte. Once they are in the bloodstream, they go to the liver or target cells of interest when the body needs proteins, right? So this is how the digestion and the absorption process of proteins works.